Hello everyone, we're going to be talking about mechanoreceptors in this lesson. We've already talked about some mechanoreceptors when we talked about somatosensory sensation associated with the skin and the sensations of touch and pressure. In this case, we're going to be talking about mechanoreceptors and how they relate to structures associated with your ear, which are dealing with hearing obviously, but also, as we will see, balance. So these are based upon having some surface structures, some cells, and protruding out of these cells, we're going to have some very, very small hairs, and these hairs are referred to as the cilia. So anything that gets the cilia moving in one direction or the other are going to stimulate these cells. That's going to generate an action potential in sensory neurons, which will send a message to the brain and provide some information related to hearing and related to balance. Probably even the simplest of organisms, the simplest of animals for sure, have the ability to at least detect gravity. And if we take a look at a fairly simple but kind of important representation here, what this is showing is kind of a fluid-filled ball of cells. So this ball of cells, we do have all around the outside the cells that are making up the structure itself, but sticking in toward the middle, we have, all of these tiny, tiny little cilia. The fluid in the middle is going to be a thick gelatinous material. And in this structure that we see here, we also have some tiny, tiny little stones. Here they call them stratoliths, but they are really calcium carbonate stones. And these stones are going to be moved around by gravity. So as gravity pulls these stones downward, it's going to brush past those hair cells, stimulate the cells, generate an action potential, and send a message to the brain, providing some information about the direction of gravity and gravitational pull. These are the structures associated with the ear, and these ones are primarily with the processes associated with hearing, although a couple of them here that do have to do not with hearing, but with balance, and they are called the semicircular canals, and a few other structures at the base of the semicircular canals that we'll talk about a little later on that do have to do with our sensation of balance and gravity. In terms of hearing, the structures that we do have here, starting with the outside, a structure called the pinna, and it is through the pinna that we're going to have the sound waves traveling. So yes, sound does of course travel in the form of a wave. This wave provides us information about the intensity of the sound, the loudness of the sound, and that's provided by the amplitude of the wave. The higher the amplitude, the later the sound is, and that's measured in units of decibels. And the wave itself also provides information in terms of the frequency. So if we take a look at the wavelength from crest to crest, the wavelength does have to do with the energy of the sound. The closer those waves are together, the higher the energy, the higher the pitch, or the higher the frequency. And whereas the amplitude is measured in units of decibels, when we talk about the wavelength or the frequency is measured in units of hertz. And humans have a fairly broad range in terms of the frequency that they could hear, somewhere on the range of 20 to 20,000 hertz. And again, that's the number of peaks that are reached. It's the number of peaks per second that are reached. So 20,000 hertz, 20,000 of these peaks reached per second. So that's a very high frequency, high energy sound. At the lower range, 20, 100, that's a very low energy, low frequency, low pitch sound. At any rate, as it is traveling through the pinna, which is collecting and funneling that sound information, it then travels through the ear canal, or the auditory canal, as it says here. And eventually what happens, as we will see, is it's going to push in on a flexible membrane-like structure. Common name for it is the eardrum, but here, and the name that you should know, it is called the tympanic membrane. So all of those structures so far, they are part of what is called the outer ear. The tympanic membrane is the entrance into the middle ear. So in the middle ear, what we do find, as it's identified here, it is still going to be air, or should be air, in the middle ear. It's in the middle ear that we have three different bones, the tiniest bones in your body. They are referred to as the malleus, the incus, and the stapes. You do not need to know those individual names, but you should know collectively they are referred to as the ossicles. Three tiny bones that you do find in your middle ear. At the other end of the last ossicle, the stapes, what we do have is another flexible membrane, and this other flexible membrane tucked in behind the stapes is referred to as the oval window. So if I can sort of draw it back in here, back in behind the stapes is where we do find the oval window. Kind of like the tympanic membrane, it's going to vibrate back and forth, and that's going to transmit the mechanical vibrations that were originally in the sound waves through these various different physical structures. So the physical structures being the tympanic membrane and the ossicles, the malleus, the incus, and the stapes. These three ossicles, if the sound is too quiet, they are going to amplify the sound. And if it's too loud, they are going to dampen that sound. There are muscles attached to these bones that do allow them to accomplish that. But they're also transmitting the information, the mechanical information, from the outer ear, and now the oval window is the entrance into the inner ear. One other structure that I'll mention here that is associated with the middle ear as well, and it is referred to as the eustachian tube. The eustachian tube is connected to your throat, and this allows the air pressure between the outer ear and the middle ear to ideally be equal. If they are unequal in the pressure, then that does create some painful sensation in and around the region of the ear. Structures of the inner ear are the ones that are really important for the process of hearing, and a little bit better as we'll, or later as we'll see, for the process of balance as well. And these structures, some of the finer ones that we will take a look at, are associated with what is called the cochlea. The cochlea is this kind of snail-shaped structure that we have here. The cochlea itself is a fluid-filled structure. It's inside of the cochlea that we have the receptor cells, the mechanoreceptor cells, for detecting those sound waves that have now been converted into the mechanical movement of the fluid inside of the cochlea. So this picture here at the left-hand side, again, it's showing us here clearly the middle ear with the three different ossicles. We can see now the oval window, the entrance into the cochlea. It has the third flexible membrane identified here as well, and this third flexible membrane is called the round window, and that is at the very end of the cochlea. So the sequence of events that are involved here, we have sound waves that are traveling through the pinna, traveling through the auditory canal, and striking the tympanic membrane. The tympanic membrane is going to vibrate back and forth, and that in turn is going to transmit these vibrations onto the three ossicles in the middle ear, the malleus, the incus, and the stapes, amplifying or possibly dampening that sound if it is too loud. The final bone, the stapes, pushes in on the oval window, and this is going to transfer the vibrations into the inner ear. The oval window is the first structure of the inner ear and the beginning of the cochlea. And then we do have within the cochlea, the fluid-filled cochlea, there are hair cells. These hair cells have hair or cilia sticking up into the cochlea, and they rest upon what is referred to as the basilar membrane. The actual sensory organ for these mechanoreceptors associated with hearing. It is called the organ of Corti. So now we've stimulated these cells in the cochlea. An action potential will be generated and a message is sent to the brain. And in the case of the brain, again, that is going to be the temporal lobe where that information is going to. Here we're just zooming in on some more highly enlarged and magnified pictures of some of the structures primarily in the region of the cochlea. So that's what we're taking a look at here. What they have done here is cut into the cochlea, and now we're looking into it, and we can see that there are these different cavities that are filled with the fluid here, here, and here. But the key is kind of what we find right along the center. Right along the center is where we have this basilar membrane, as it identifies down here, and it's sitting on top of the basilar membrane where we do have the very important hair cells with the cilia that are sticking upwards. All around these cilia, again, it is going to be fluid filled, and we do have one other structure here. It's a flap of tissue over top of those cilia, and it's referred to as the tectorial membrane. So you can sort of think of these hair cells, the cilia themselves, as rubbing up against the tectorial membrane. Fluid within the cochlea is moving to result in that movement, and that's what's going to generate action potentials, sending the message through the auditory nerve and to the occipital lobe of the brain, or rather the temporal lobe of the brain. And over at the left-hand side, we do have an electron micrograph image that is showing a picture of these individual hairs, or rather a group of these hairs, sticking out from some of the hair cells that we do see here. Now, many things can damage these hair cells. Exposure to loud sounds, especially over prolonged periods of time, can cause damage and can cause damage to specific regions along the length of the cochlea. So let's take a closer look at the cochlea. Again, it is a fluid-filled tube that is coiled up, and that's why it's sort of referred to as this snail-like coil. And of course, it is part of the inner ear. Inside is where we find the receptor cells sitting on top of the organ of Corti. We'll see a picture in a second, but hair cells are shorter at the beginning. These ones are shorter, they're stockier, it's a little bit more difficult to move them, so you need more high energy, high frequency, high pitch sounds in order to move those ones. So it is the beginning of the cochlea then that is going to be detecting the higher pitch sounds. Toward the end, the hair cells are going to be longer, stringier, not quite as difficult to move. Lower energies are sufficient to move them, so these respond to the lower frequency or lower pitch sounds. Enlarged picture here showing what we saw before, and again, this is the region that we're kind of focusing on for hearing. So we do need to, again, get the fluid moving through these canals, and all of that is going to be initiated by the original sound waves that are traveling in and pushing in on the tympanic membrane. As we saw before, the hair cells on the basilar membrane are rubbing against the tectorial membrane and the auditory nerve sending messages to the occipital lobe of the brain. And that last window, the round window, is the structure that's going to eventually dissipate those vibrations when they do reach the end of the cochlea. This is the snail-shaped cochlea that we're taking a look at here. You certainly don't need to remember any specific frequencies, but I did mention that the beginning of the cochlea responding to higher energy sounds. Toward the end of the cochlea, it's responding to the lower energy sounds. And on the right-hand side here, they've just kind of uncoiled the cochlea. And yeah, indicating at the beginning, high frequency, toward the end that we see here, the lower frequency sounds, and we can tell the difference in the frequency or the energy if we take a look at the distance between the crests, the distance between the waves. The closer they are together, the higher energy they're going to be. Again, just a more magnified picture again, zooming in even further and taking a look at the organ of Corti. Hair cells, cilia sticking up, rubbing against the tectorial membrane as vibrations are passing through the fluid-filled cochlea. This is a section of the cochlea showing the rows and rows of the cilia of the hair cells. And again, they can be damaged. It looks like down here, possibly, these ones in fact have been damaged, possibly as a result of loud sounds, exposure to loud sounds. This one here is showing that in fact they are separate receptor cells, just like we saw with the photoreceptors in the eyes. So we do need to have a connection between these receptor cells in the organ of Corti and the sensory neurons that are part of the auditory nerve. So there will have to be the release of a neurotransmitter. And what this picture here is showing is that initially we have the hair cells that are not moving. If you get the hair cells moving in one direction, that might generate an increased rate of the action potentials. And if you get the hair cells moving in the opposite direction, it would give us, well, the opposite response, less neurotransmitter released and a lower frequency of action potentials going toward the brain. That is pretty much it for hearing. For balance and equilibrium, we'll see that there are some similar processes, and similarities do have to do with the hair cells, with the cilia and the fluid-filled environment that those cilia are sticking into. And in this case, the movement is going to provide information related to your balance and your equilibrium. Major structures involved here, I pointed out on an earlier slide, what are referred to as the semicircular canals. So they are canals that form, well, kind of like what I'm drawing here, sort of a semicircle. But what you do have are these semicircular canals that are oriented in three different orientations. So this one here, we could perhaps say is the Y orientation. You would have another one that's going in the X orientation. And another one, if I can draw it here, that's going in the last orientation, which we'll call the Z orientation. And what this allows you to do is respond to movement, yeah, in any direction, whether you're going front to back, side to side, or spinning around. These semicircular canals allow you to detect movement in any of those different directions. At the base of these canals, what we do have are structures that are called the ampulla, and it's inside of these ampulla where we do have some hair cells, where we do have these hairs that are sticking up into the fluid. So as the fluid does start to move through one of the semicircular canals, let's say this red one that I've drawn here, if it starts to move through in the direction that I'm showing, what we're going to have at the ampulla at the bottom are going to be some hair cells, some hairs that are sticking up, and that's now going to get those hairs moving in one direction. That's going to generate an action potential, and as I've been saying over and over again, send a message to the brain, going to the part of the brain that's dealing with what's called dynamic equilibrium, the movement of your body, and provide information about movement in any direction that your body might be taking. So much better picture here than the one that I sketched, but again we can see that we have these semicircular canals in different orientations. So we could maybe call this one here the X orientation, this the Y, and this one here the Z orientation. Ampulla we can see at the bottom of each one of these semicircular canals, and that's where we would find the hair cells that are sticking up into the fluid going through the semicircular canals, which is continuous with the fluid in the ampulla. At the left-hand side here, it is showing a portion of one of the semicircular canals, in fact, the base of it. So if I were to draw the rest of it, it would look something, well, perhaps like this, looping up. So again, the key with this is if we get fluid going through the semicircular canal, if I'm doing a cartwheel in one direction, the fluid will start to go through this semicircular canal and it'll push the hair cells in one direction. If I do a cartwheel in the opposite direction, well, fluid will then travel in the opposite direction, move the hair cells in the opposite direction, and when that information goes to the brain, it will be interpreted and processed and provide information about my body movement. The other picture that it's showing on the right-hand side here, this one has to do more with what's referred to as static equilibrium. So providing information about your body when you're at rest rather than moving, like we see with the semicircular canals, and also your body position relative to gravity. As we can see here, this is a very similar situation to that first picture that I showed you, because you do have some structures here that are tiny, tiny little stones or rocks, calcium carbonate rocks that are located in some other fluid-filled structures. But in these structures, once again, we have the same sort of arrangement. We have some hair cells, we have some hairs that are sticking up, some stereocilia, and as these tiny, tiny little stones are moving around within the fluid, they brush past the hair cells and they provide some information about your body position and body position relative to gravity. So these different structures, they are referred to as the saccule and the utricle. So these detect gravity and linear momentum. And I mentioned these tiny, tiny little stones, calcium carbonate stones, that are going to be moving around and pulled in the direction of gravity. So the picture that we see at the bottom here, initially in this picture, the stones would be pulled down, brushing by the stereocilia, providing information that this person is in an upright position. When they do bend over, the stones are going to be pulled, well, in a different direction, brushing by those hair cells, providing a different frequency or pattern of action potentials, which again is sent to the brain and interpreted as the person is being bent over in the forward position. That is it for the hearing and balance. Proprioception was mentioned before. Just one final comment about proprioception. These deal with another kind of mechanoreceptors that are located in locations like your muscles, your tendons, your joints, and they provide very important information about the angle of your joints, your body position, and the movement, angle, orientation of the limbs that you have.